Welcome back, everybody. I'm Dr. Andrew Heyman, the director of the Fellowship for Integrative Medicine here at George Washington University, and I have the privilege and the honor to introduce Dr. Frame. She has accomplished so much in her career. I have to decide which bio, and I think this one it best encapsulates. So Dr. Frame is Chief Wellness Officer of George Washington University Medicine and Director of the Resiliency and Wellbeing Center. She also serves as Executive Director of the Office of Integrative Medicine and Health and Associate Professor of Clinical Research and Leadership and Physician Assistant Studies. As co-founder of the Resiliency and Wellbeing Center, she's advanced resilience, whole person care, and well-being initiatives across GW and beyond. With extensive experience in biomedical research, Dr. Frame bridges nutrition and immunity through translational research. Her interests include the microbiota, gut-brain immune interface, malnutrition and obesity, vitamin D as an immune modulatory hormone, research ethics, science communication, resilience, and whole health. She consults for and serves on global healthcare and biotech boards. In 2021, she was named among the top 100 healthcare leaders by the International Forum on Advancements in Healthcare. Dr. Frame graduated with distinction in biochemistry from Mary Baldwin College, now Mary Baldwin University. She earned her MHS in molecular microbiology and immunology and her PhD in international health, human nutrition from Johns Hopkins Bloomberg School of Public Health. While pursuing her PhD, she gained expertise in obesity and metabolic health through work at Johns Hopkins Center for Bariatric Surgery. I read all of that and I think you probably listen to that and think, gosh, did I really do all that? And, you've already had an amazing career. So I know everybody's excited to hear what you have to say.
Sorry about that. My Zoom meeting controls went away and then I couldn't figure out how to unmute myself. I am back and very glad to be here. Okay, so what we're going to talk about today is just kind of an extension of what we've been talking about, but I think maybe putting it in a slightly different context, which hopefully will be helpful for both clinician and individuals alike. My conflict of interest slide, nothing affects the contents of this talk. Okay, so we're going to talk a little bit what I think of as kind of as the missing piece of the fasting discussion so far, and sort of why fasting works on a biological level in a bigger sense as opposed to more the nuanced sense that we've been talking about so far. So I want to put this in context. Here's a nice little visual here, sort of the beautiful symphony that is the human body. And we have all sorts of inputs, like for instance, the light dark cycle that set our circadian clocks. And another one of these is food intake. And so that's what we're going to focus on here. Why is food intake important for our circadian rhythms? And really, fasting, while it does have caloric restriction, and we've talked about all the benefits of that, it's not just about that. It's also about synchronizing our biology. And this involves the gut microbiome, the liver, and metabolic processes, which we're going to dive into here. And my hypothesis is that the real power of fasting may actually be how it interacts with our biological clocks as opposed to the simple caloric restriction. Okay, so the gut microbiome, we're going to start here. It's one of my favorite topics, and it does have its own daily rhythm. But it doesn't see the sun, right? How does it know whether it's day or light? Well, I'm going to tell you that it's a fasting regulated ecosystem. So the gut microbiome has this daily rhythm, and as part of that rhythm, you actually see a shift in bacterial species depending on the time of day and the fasting state. And it's really that fasting that's going to shift that composition and thus the function of the gut microbiota. What are they doing at certain hours of the day? You're going to see certain bacterial species are actually going to peak at different times of the day. So they're not all changing in the same way. Some are going to go up, some are going to go down, and that's going to maybe reverse in other parts of the day. Some really thrive in that fasting state. And so for them, it's important to have some fasting period of time to kind of grow up and become strong. And when I talked about the microbiota, I talked about what are they doing, right? They're making metabolites. And metabolites like short chain fatty acids and bile acids and polyamines have all been shown to fluctuate, one according to who is there, but because who is there is shifting over the day with a daily rhythm as well. So there's actually a daily rhythm of these metabolites in addition to the different bugs that are actually there in the microbiome. Oh, well, this is new. Didn't know I did this. Okay. Um, so we've got different ways of thinking about fasting. And what we've really talked about was we talked about how we're either eating or not eating. But for this talk, we're going to focus more about like when are you eating? Are you eating at during your active period? And if you're eating during your active period as a human, that means during the daytime. We would call that aligned eating. So that's aligned to your natural eating cycle. We would typically eat during the day. However, if you're a mouse, your active period is at night. And so thus aligned eating would be at night. Now, if you eat when you're not typically active, so in the middle of the night for a human or in the middle of the day for a mouse, that's misaligned eating. And misaligned eating can just be sort of late night meals, or it could even be an erratic schedule. If you don't have a regular rhythm to when you eat, that can actually disrupt your body's clock because it's not really sure when to expect food. And that changes how many things are functioning in the body, not the least of which being bile acid release from the liver, which we will talk about. And because you have this misaligned eating, you actually see a disruption in the balance of the gut microbiome. What you see is a promotion of inflammation and metabolic disease. And what does that actually look like? So if we look over here, we could see in the fed state, there's a potential for imbalance, right? You could be overfeeding or you're really skewing things towards the microbiota that don't flourish in this fed state. Remember, I said some people actually do better in the fasting state, some of these microbes. And so you could potentially be having an imbalance if you are only in the fed state. And what we typically see is carb and fat digesting bacteria are really going to grow up during the fed state. And none of this is bad. It just has to be put in balance with the fasting state. So what happens during the fasting state? You actually see more of these short chain fatty acid producing species, which if you listen to my previous Sun Symposium lecture on microbiome, you'll know is one of the key elements of linking the gut microbes and fiber to health outcomes. And so short chain fatty acids are something that is produced by our gut microbiome when we eat fiber. And it also is not only important for them, obviously they're metabolizing it, but these short chain fatty acids are actually fuel for the colonocytes in our gut. So the cells of our gut cannot function without these short chain fatty acids. And so here's a reason where the fasting state could be really important. It's a time of repair, right? You have the colonocytes, they're uptaking these short chain fatty acids, they have fuel, now they can repair themselves, they become strong and prevent leaky gut. We'll talk a little bit more about that, but that's one of the really key things that why it has to be a balance is because you have to both feed and bring nutrients in, but you also have to repair. And like I said, some of the elements of the microbiota, certain taxa or phyla, are increased during the fasting state. So it is really a balance here. Akkermansia is one you may be familiar with. So one of the stars is Akkermansia municipala. That increases during fasting. So if you are never really in a true fasting state, you will have very low Akkermansia, and that has been linked to negative health outcomes. So I will add that that is very non-specific at the phyla level. We're talking about very broad groups of bacteria. Okay, so we're going to walk through a couple of studies. This is the first one. And the title is Diet Diurnally Regulates Small Intestinal Microbiome Epithelial Immune Homeostasis and Enteritis. So this is an animal model. We're going to look at some animal model studies. And this is actually their visual abstract here. So each one of these panels we're going to walk through individually. So we know that certain cells are what we call light entrained, right? So light is telling them it's active or inactive time period. And that's really important for things like the brain, the suprachiasmatic nucleus in the brain. But like I said, the gut's not seeing that. So what is causing the rhythmicity in the gut? Well, what we find in this animal model, and I will show you it in other models as well, is it's nutrient intake. So if we have low nutrient intake, we have sort of the equivalent of the dark cycle for humans. And then if we have high nutrient intake, then it goes to this active state, right? And so we have this up and down with fed and fasted, daytime and nighttime. And that is really what's signaling to a lot of the elements that don't have this light section as to whether it is day or night. And so really those gut rhythms are depending on meal timing. And so many of you have probably experienced, if you travel, particularly if you travel around the world, you get jet lag and it's terrible. And those type of symptoms are what we are talking about when we are talking about misalignment, right? You are misaligned when you have jet lag. And one of the ways to prevent jet lag, well, there's many ways, but one of the ways is to try and time your meals more to the area you are trying to get to, right? So wherever you are trying to move your cycle to time your meals on that time, and that will help with jet lag. Same thing with light, getting light exposure on what you are trying to move your time cycle to. Okay, so that's number two. And because of that, we see this change in the microbes that are present in the microbiome. So the composition of the microbiome also shifts with day and night. And we'll talk a little bit more about that in detail. The next one is that because you're having this shift in, let's say it's active or inactive period, what else is happening is that it's actually triggering a change in the microbiome-immune reaction. So the way that the gut microbiome is perceived by our immune system. And so you see a different activation of the immune system. And so during well, this is actually reversed because this is mice, but during the inactive period, during the time of rest, the immune response actually gets higher. So the immune response over here is actually getting higher, and then it gets lower during the day. And we'll talk a little bit about that in detail. But essentially, it's sort of this balance, right? You don't want to have too many microbes, but you don't want to have too few. And so having this oscillation allows a maintenance of balance. And what you also see is a change in a reaction in terms of what cytokines are actually produced. So IL-10, for instance, is what we typically would refer to as an anti-inflammatory cytokine, and it has sort of the opposite rhythm to immune sensing. So if immune sensing is peaking during the inactive period, IL-10 is going down, and vice versa. So here we actually want to have the immune system reacting, so we're turning down this anti-inflammatory IL-10 to actually spark an immune response. And what we find is that misaligned meals are actually disrupting this. They're disrupting this, and this cycle is lost. And when you take an animal model that has had this disruption through misaligned meal timing, it actually increases their susceptibility to enteritis. So they're more prone to this pathogenistic outcome simply by changing the misaligned meal timing. Everything else is held constant. So light's held constant. All of the things that we maybe would typically think of when we think about circadian rhythm are held constant, but food is changed. But the best thing is when you bring back them into aligned meal timing. So you shift away from the misaligned and you go back to an aligned meal timing. You actually can reverse that. It promotes gut resilience. So it's something that is reversible. And again, any of us who've ever been jet lagged, you understand it is reversible. It is very, very uncomfortable when you're experiencing it. But the problem is if we have prolonged exposure to that. And so we'll talk a little bit about that later. But first, let's talk about what else they found in this study. So they looked at gene expression. Here's just a little bit of a 101 for those of you to make sure you know what we're talking about. So we have DNA, right? DNA is basically the template from which we make things. And so we transcribe the template. Here is the message. And then that message is translated into proteins, which actually are doing things. They're the enzymes, there's the structural proteins. And so this is what we're talking about when we're talking about gene expression, is this process. And in this study, looking at the mice, and they actually looked at the gene expression in the small intestine, so locally in the small intestine, they found that over 1,600 genes showed changes in their expression patterns that were cyclical due to circadian rhythms from, from meal timing. So a diurnal variation that was indicative of either whether it was aligned or misaligned meal timing. It was following that pattern. And because of that, if you are controlling the feeding time, you're essentially controlling the peak expression of these genes, all 1,600 of them. And here are some example genes just to get a feeling. So Reg3 and DEF-A, which are antimicrobial peptide related genes. So, okay, what are we talking about here? Antimicrobial peptides are part of the immune response. So you could see why it would make sense for this to be on a fed fasted timing, because we want a stronger immune response while we're fasting because we're doing that repair and recovery. And we want a lower immune response when we're feeding because it's more important for us to be processing the food and getting the nutrients from the food. Another one is claudins and other tight junction proteins. So I kind of alluded to this, right? Leaky gut. The reason that a gut can be leaky is because those tight junctions between the colonocytes, they're weakened for one reason or another. Maybe the colonocytes don't have enough fuel and they can't repair themselves, and those junctions become leaky. And they're important for that barrier function and protection from pathogens like enteritis. And so what happens is when you have feeding, it actually suppresses the tight junction, not feeding. When you have misaligned feeding, it suppresses the tight junction proteins. So if you're feeding, well, actually, both is technically true. That's why I'm struggling here. So let's talk about just feeding generally first. So when you're eating food, you want to get the food from the inside of the gut to the outside of the gut, right? You want to be able to absorb it. And so there is some downregulation of the tight junction proteins to facilitate that. That being said, you also want to then turn that off, right? As soon as you've absorbed as much of the foods that you need. And so that's important. But then misaligned feeding is sort of causing more of a general downregulation of tight junction proteins, what we would traditionally think of as a leaky gut. So it's getting turned on generally. So you always have this downregulation of a tight junction protein instead of it going up and down with meals in a more health promoting way. Okay, so now we're going to look at this mouse in terms of its feeding. So I already told you the inactive phase is the daytime, the active phase is the nighttime. I know it's kind of confusing because it's the opposite of us. So we're going to just talk about aligned versus misaligned. So if we have these aligned feedings, what we saw was oscillations in the microbial composition. So we saw things like lactobacillus or Bacteroida changing with the inactive versus active phase, depending on whether they were being fed or not. And misaligned feeding basically undid a lot of that, right? So you typically would have a microbial rhythm, but when you have misaligned feeding, that's kind of flattened. It's sort of changing the difference between the fed and fasted state because they're not happening at the right time. What else did we see? We saw a drop in diversity, which if you know anything about my talks, you know that diversity is the sort of most agreed upon marker of gut microbiome health, which we can't even define what a healthy gut microbiome looks like. But this seems to be the one marker that sort of generally is health promoting. And so we don't want diversity to go down. That's a negative thing. And then metabolites also lost their rhythmicity, including short chain fatty acids. So again, there's not as much of a difference between the fed and the fasted state. It's all kind of blunted. And the same thing then with the immune activity and the ability of the immune system to actually control that microbial growth in a balanced way is also blunted. So it's still there. It's not completely inactivated, but it's just not as strong as it needs to be. So essentially, these feeding fasting cycles, they're gonna train the microbiome, right? They're gonna say to the microbes, this is what you should be doing now, this is what you should be doing later. And it's this cycle that they kind of get into. And this is why it's important to have rhythmicity of eating, right? There's an on period, there's an off period, and having it generally be regular is really important for them to kind of get into that cycle, or resetting the cycle if they are off that cycle. And the microbes themselves are actually important for training the immune system. So this is just an example here of how the microbiome can interact with the immune system. And this is what we call immune education. So the majority of your immune system at any one period of time is residing in your gut. And that is because the gut for the gut micro or for the immune system, it's basically like grad school. They are going to like learn higher level information. They are learning whether this is a state that is generally inflammatory and they should be ready to fight, or if everything is kind of normal and they're just out there looking and surveilling and making sure that everything is okay. So that's what's going on here. So you can see if you have an alteration in these microbes and the response that they're being the message they're giving to the immune system, that it could be detrimental, sort of largely speaking, not just in the gut, because the immune system is then going to travel over across the whole body and use the information that it learned in grad school throughout the body. This, however, is still being elucidated. So this is kind of early research. And there are certain cells that have shown to be particularly involved here. And I'm not going to get into details on this other than this one particular cell type, it secretes antimicrobial peptides. So that's something we're going to hit on time and time again during this talk. You will see that comes up many times. And there's actually a rhythmic release of the granules that hold the antimicrobial peptides in these cells, and it's triggered by feeding. So the only way to alter that timing is by having a fed state and a fasted state and having it be regular and rhythmic. Similarly, goblet cells, they're mucus producers. So this green area here, this is mucus. It's a really important part of how the gut microbiome functions. It keeps enough distance away from the immune system that they're not constantly on alert and being like, oh my gosh, they're invaders. And so it's really important to have enough mucus. And we found that in these mice, when they were misaligned, the mucus producers suffered. They did not have the same rhythmicity as you would expect to see. And so there's alterations then in that mechanism as well. So here's sort of a general idea of what this model looks like. So they used a DSS-induced colitis. So this is basically they're giving a chemical to induce colitis in this mouse model. And sort of generally speaking, what happens there is you see damage to the colonocytes, right? You might have a little bit of leakiness, things are getting through. The mucosal layer is gone. See, over here's a nice healthy mucosal layer. There's no mucosal layer here. So basically, they've kind of undone a lot of the control that is typically there between the gut microbiome and the immune system, and things are really off the rails. And what happens is they develop colitis. And when you have this model with misaligned feeding, you're basically adding some gasoline on that fire. So what do you see? You see more severe enteritis. You see a higher epithelial damage, so more of this damage with misaligned feeding. And then you see immune dysregulation, which sort of makes sense, right? Like they're here at university and they're trying to learn, but basically they're in the middle of a war and they aren't really learning what they need to learn. And so their response is often inappropriate. But the good news is they were actually able to rescue them with time-restricted eating. So if you get them back onto an aligned feeding regimen with a restricted window, that actually restored that protective gene expression, those 1,600 genes, and the microbiota rhythmicity. So it came back, right? The increase in certain bugs and the decrease in the others, and then the flow of short-chain fatty acids and other metabolites is able to be restored when they implemented time-restricted, or you would call it time-restricted feeding in mice. It's time-restricted eating in humans. But it's the same concept, right? So this could be a mechanism for overcoming guts that might have a pro-inflammatory status. This may be beneficial, but obviously we're talking about mice here, so this is still early phases. All right, the next study, this is another mouse model. So again, we're in the early phases, but I think this one's gonna blow your mind and it's gonna be really worth it. And here we're looking at sort of the mechanism behind feeding dependent regulation of the intestinal barrier function. Okay, so this study replicated some of the findings of the previous one. They found that the gut microbiome composition did have a diurnal variation, this rhythmicity, that was driven by fed and fasted states. So again, it's in mice, but I will tell you that later we're going to talk about that it's not just in mice. but it's just much easier to study these things in mice sometimes. And over here is actually the data for that in case you are interested. So they have a number of different types. Basically, fasted is here and fed is here. And you can see really there's a pretty clear difference between the fed state and the fasted state. There's even more so over here. And this is with small numbers. These are early studies, but I think the mechanism is so strong behind these, it would be surprising if we aren't able to continue to replicate this. And so this is the part I'm really excited about. This is actually microscopy of the gut. So these little buds here, these are the villi, the little fingers of the gut. Well, go back to one of those pictures, right? So they've got these little fingers here, these projections. And what that does is it gives more surface area for the gut to absorb nutrition. So it's a really important indicator of the gut, by the way. And so here we've got these little fingers, and what's going on with these little fingers? Well, they have a little bit of these bacterial threads. These are basically chains of bacteria. There's a little bit of them here, and there's lots of them down here, and they're much longer. And the only difference between these two groups is this group is fasted, and they have the stubby chains, and this group is fed, and they have the long chains of microbes. And so this is really showing us what we're talking about, right? Like the immune system during the fasted state turns on and it knocks down the microbes in the gut microbiome, kind of controlling them, saying, okay, that's enough growth for one day. And then when you go back to the fed state, they can grow back up again. And it's this cycle of up, down that's really important for helping to control, because you don't want too much of a good thing, right? Like if these guys just kept growing and kept growing, then you'd have microbial overgrowth, which is a known problem. So we don't really want to be promoting that indefinitely. So it's important to have a fasted state. And how does this work? Well, it works through the immune system, not surprising. And IL-22 is one of the big ones. When we feed, so we get out of the fasted state, IL-22 production is actually reduced. And when it's reduced, that causes a decrease in gene expression. So this one should look somewhat familiar. Hey, it's an antimicrobial peptide. So we're having the peptides that are sort of like antibiotics for within the body, right? So they help reduce the count of these bacteria, and it's decreased because we're feeding and we're less concerned about knocking down the bacteria. Right now, we want to do more of the absorbing nutrients. And with that, you see this change in morphology, right? So you see it going from stubby to long, and altered composition as well, which we talked about earlier. Now, what's really cool is they've got these two conditions here. So this condition is the one we just talked about. The only thing that's different here is fasted versus fed. Over here, they delivered an anti-IL-22. So they're sort of blocking the IL-22 production without the use of feeding. So it should be somewhat in the middle, right? So it's sort of like here we have a fasted state, but you're blocking IL-22, so it's not as much of the fasted signaling is getting through. And so what you see is a little bit more growth here, right? There's a little more growth than there is here. And down here, it's a little hard to tell because there's already so much growth here, but there is a little bit more growth here than there is here as well. So the IL-22 really disrupts the ability of the immune system or the anti-IL-22 disrupts the ability of the immune system to control microbial growth. Okay, so what happens when we feed? There are actual mechanisms behind this. So enteric neurons, these are basically neurons, right? Brain cells that are in the gut. And it's part of the gut-brain axis, the communication that the brain has to the gut and the gut has to the brain. And when we feed, these neurons actually activate a specific pathway. And what that pathway does is it inhibits that IL-22 production. So this is the mechanism behind IL-22 working. And IL-22 then reduces the antimicrobial defenses. It also increases greater nutrient absorption, which I've alluded to, but we haven't specifically mentioned. And in this particular study, they specifically showed that lipids were poorly absorbed when you had IL-22 production, or when you had decreased IL-22 production. It also increases the enteric pathogen risk, right? So that's why we're talking about enteritis. There's a higher risk for developing gut inflammation and pathogenic infections. But it's important to balance this. This is all about balance, right? So we've got this neuron activation, right? That's because we just fed. We also have a change in the immune response. So IL-22 is turned down. The ILC3 cells, these are immune cells, are actually changed the way they work. The microbial control is loosened, and that increases the pathogen susceptibility. And so, but we also have this trade-off here with nutrient absorption. So we want to loosen the microbial control so we can enhance our nutrient intake. And so it's actually a trade-off between defense and absorption. As with many things in the body, we sort of need to do one or the other. I like to tell people that you can either digest or rest. Right? You need to eat your food, digest it, and then go to sleep and get a good night's sleep so you can switch to the repair. And this actually is very similar, right? You can't do both. You can't be feeding and absorbing nutrients and be repairing and knocking back things and having the immune system actually respond. And so it's about this balance. You don't really want to be always fasted, but you also don't want to be always fed. And having this sort of ebb and flow is really natural to the way our immune system functions and the way we control our gut microbiome. Okay, so let's just talk about in brief, what does this mean for TRE? So in health, what we find is that it increases microbial diversity. We see beneficial metabolites are produced. It seems to be beneficial for the immune system. It sort of works with the immune system. And then it's also beneficial for metabolism, which we're going to talk about a little bit more right after this. But first, we're going to go to the liver. Liver, also a fasting regulated system. So this is going to be set up very similarly. When we have a fasting state with the liver, what happens? It's detox enzymes are activated. It moves to a fat breakdown priority, so you're burning fat, and it's releasing glucose or causing ketogenesis. So obviously in the feeding state, it's going to be very different, right? So those detox enzymes are inhibited. It's not time to repair. It's not time to clean things. And we're going to be creating fat. It's time to store, right? We just fed, let's store fat. And the same thing for glucose. We're going to store that for later. So what happens in this process? So if we ingest carbohydrates, we have an insulin response that involves the pancreas, which by the way is also a fed-fasted circadian rhythm. So we'll talk about that. Then you have the uptake. The liver is actually utilized, so the liver is activated. And here's really part of the reason that the liver is so important here, because the liver becomes active in response to food, right? It doesn't come active in response to sunshine. So when you are feeding your body, your liver says, it's time to work. And so this is really one of the key here. And then you can, use your muscles and your fat tissue and everything else around it. But it's this key here that the liver needs to be utilized and it needs to be awake. And similarly, you might be having ketosis. And so for those of you who don't really know what ketosis is, basically it's a way to take fat and turn it into a form of fuel that can be used by the brain, because the brain really is more of a glucose user. And the only way it can use fat is this ketone synthesis, which occurs when we are fasting. And so typically when you are fasting overnight, you will enter ketosis and your brain will be running off these ketones, which allow the brain to function in a very different way. And it's switching to that repair mechanism, which we'll talk about. However, we have this issue here with overnutrition. So a lot of, a lot of people in our world don't ever fast, right? We always have food around us. It's very difficult to fast. Many people even get up in the middle of the night and eat. So now you're not even having that overnight fast. And it can really disrupt the whole system. This continuous fuel delivery, it can actually alter the mitochondria, the way the reactive oxygen species are produced, our proteins, and even lead to cell injury. And we'll talk a little bit more about that. So what are the metabolic benefits here in fed versus fasted states? So if we maintain the fed state, what's the pros? Well, we're going to have rapid energy, stable blood glucose, the tissues can grow. We're in that strength building stage. We won't have hypoglycemia. We should have metabolic flexibility in that we've kind of got whatever we need sitting around to create whatever components we need. What are the cons? The potential for overconsumption and possible weight gain, right? If we're constantly eating, it's a little harder to control. This is why we're talking about TRE as being a way to restrict calories. There is potentially a risk for insulin resistance, obviously, depending on what type of food you're eating. There is the potential for nutrient imbalance. Are you having too many of a certain nutrient, and then you're going to lose some of these fasting benefits. So, okay, so this is one of the big reasons why we don't want to maintain a fed state is there is a reason to be fasting. What goes on in the flip side in the fasted state? So you do have energy, and that's thanks to our stores as well as ketogenesis. Brain function support. So the brain is getting the ketones that it actually really enjoys taking a break. It's like practicing metabolic flexibility, and it's allowing itself to repair. It's also important for muscle protein. So this can be quite preservative. We don't use the muscle protein as fuel as long as we have enough ketones. Regulates inflammation, produces oxidative stress. But there's cons to that too, right? So we might have energy dips. We could have hypoglycemia. There's definitely a risk for nutrient deficiencies if you are fasting too much. There could be an adaptation period where maybe you're not feeling so fantastic at first. It's also not suitable for everyone, which we're going to talk about in the next talk. And there is a risk of muscle loss if it's not managed appropriately, but really we're talking about extended fasts there and less so time restricted eating. So I think I alluded to this. It's about balance. It's the yin and the yang, the fat and the fasted state. You're either having protein synthesis, ketone production, a prolonged fasting, or chronic insulin elevation. None of these are particularly good, right? We don't always be in protein synthesis even. Like we need to move from one to another and have breaks, and having that balance is really what is key. So what happens to the liver when we're fasting specifically? So we start fasting and it immediately begins to adapt because the liver is very adept. It will switch to ketones if needed. It will also begin to cleanse itself if we have longer fasting periods, that autophagy that we've been talking about. And then it switches to the fat oxidation, right? So it's actually breaking down fat for energy. It will also be detoxing itself. So that's really important. And that mitochondria undergo that alteration in the way that the liver is utilizing energy production will happen sort of the later phases of fasting initiation. So if you start fasting and then you maybe get down here to okay, we start to dip into some of our ketones, but then you pop back up, right? That's what we're talking about. You don't get down to the autophagy and any of the other benefits. There is a period of time you need to be fasting in order to receive these benefits, and Dr. Peterson talked very nicely about that. So what is going on with the liver? So it's got circadian rhythms. It has core clock components. These are actually regulators of that cycle. And there's an interaction with the liver and the brain, not surprisingly. They're talking to each other, saying, what's up? What time is it? Is it eating time or is it sleeping time? And that actually changes the feeding behavior, and that can be disrupted if we are not on an aligned feeding schedule. So a little introduction here to my favorite friend, the vagus nerve. I love this photo because you can see how this nerve really, it goes everywhere. It's called the Great Wanderer. That's what vagus means because it goes all over the body. It's really important for sensing. It goes into the gut, it goes into the liver, the pancreas, all over the place. And in this study by Woody et al., they showed that in mice, the vagus nerve is actually part of this control of the liver food intake and that brain connection. So if you've got a mouse and it's just eating normally, the liver's on a normal circadian rhythm, it signals to the vagus nerve and the hypothalamus, and everything's good. We're all in alignment, right? The brain clock is the same as the liver clock. But if you have a dysregulated or a misaligned eating behavior, what happens is the liver clock is on a different time than the brain clock. And that dysregulation makes a lot of the negative effects happen, is that misalignment between the brain and the liver. It's the liver's trying to do one thing, the brain's trying to do another. Like the brain's trying to rest and repair while the liver's trying to make new proteins or all that sort of stuff. And what is really interesting is you can actually what we call rescuing this phenotype by ablating or dissecting the vagus nerve. So you're cutting the connection between the liver and the brain, and it restores some of the negative effects in mice at the very least. So again, the vagus nerve and that connection with the brain is going to be part of the effect here. And it's the disconnect between the two that can be quite problematic. Okay, so let's talk a little bit of the gut-liver axis. Why are they connected? So we've got the irregular eating. This disrupts the gut microbiota. That also changes the bile acid signaling, right? So the liver is producing bile acids in a way that is not typical or aligned with our cycles. And this increases our risk for metabolic disorders like insulin resistance, fatty liver disease. That is what we're calling a disrupted gut-liver axis. Okay, so how do you reset this? What is going on? Well, fasting is what we'd call as a fancy term is a Zeitgeber, which basically just means it's a cue that resets the circadian clocks. And there are many different kinds of Zeitgebers, right? There are ones that are we call first order, which is like the sun and temperature, and second order ones, which are like food intake, right? So they are different and they interact differently with the body. Light is for the brain. Food intake is for what we call peripheral clocks, the liver, the gut, pancreas. These are all peripheral clocks. They're all driven purely by food intake. And so fasting is a Zeitgeber. It's a way to reset that chronobiological clock. And thus, it's a tool. It's something we can use to realign our clocks with day-night cycles. If we are eating within aligned windows, that should, in theory, produce all of these benefits, right? We eat in an aligned window that synchronizes our clocks, that enhances metabolic efficiency, and may also have some of these benefits of improving glucose regulation, supporting immune function, and optimizing health. And in theory, it's as simple as not eating when it's nighttime. It is a little bit more complicated than that, and there is more nuance, and it depends on the person. But that's a good rule of thumb: is if the sun is out, it is okay to eat. So if you're hungry, it's always okay to eat. Okay, so how do we weigh the pros and the cons of time-restricted eating? So pros, it improves metabolic efficiency, glucose regulation, it can enhance immune function, it's getting us on a good circadian balance. It's probably one of the biggest pros. And then there's no calorie restriction needed, right? We don't have to count calories. We've already heard that today. The cons, potential social limitations, right? Are you able to go out for dinner with your friends, or maybe you can convince them to have dinner a little earlier? There may be an adaptation period where you're kind of slowly moving towards a smaller and smaller window. If you're not typically used to doing this, that would be my recommendation is don't just change yourself abruptly because you're going to shock the system. It's possible you might be hungry and maybe the window is too long for you if you are having that experience, at least at this moment. That limited eating window can be psychologically challenging, which I'm hoping the last talk will talk on a little bit. And it really isn't suitable for everyone. So I wouldn't use this as a blanket tool. Okay, what else is going on? The immune system and brain health. So I alluded a lot to the immune system. We're going to fly through this because of that reason. But essentially, aligned feeding promotes a healthy immune system. And disrupted is disrupting the immune system. So you really need to keep that in mind as we're moving through some of these details. Okay, so when you have aligned feeding, what happens? We have an enhanced immune response and effective surveillance. What does that mean? What is another way of thinking of that? It's promoting homeostasis. And during homeostasis, we have immune surveillance and a robust response if triggered. So it's kind of just there and waiting and ready to go. But it's not turned on. It's not chronic inflammation, right? So ideally, this is what we want the immune system to be in. So aligned feeding promotes really an ideal state of the immune system. And it also supports immune cell timing. Well, what does that mean? So there's actually circadian-driven migration of certain immune cells. So neutrophils, macrophages, and T cells are triggered to move throughout the body by this circadian rhythm, by this fed-fasted pattern. And because we are aligned and our immune system is in this homeostatic way, that reduces chronic inflammation, right? It's not just willy-nilly turned on as it would be in chronic inflammation. And because of that can aid recovery, right? You're in a rest and repair phase instead of a inflammatory response phase. Not surprising, misaligned is basically the opposite, right? So we're disrupting that circadian rhythm, we're impairing the immune response, increasing infection risk. It promotes inflammation-related diseases, and there is some indication that tumor formation risk may increase by misaligned eating. All right, what about the brain? So the brain, we already alluded to. Autophagy is really important in general. It's really important in the brain too, right? So we're having cellular cleanup, reducing neuroinflammation. And if you know anything about neurodegenerative diseases, these are both really important in there. So I'm not saying that fasting can necessarily treat neurodegenerative diseases, but it may be something that is at least able to slow or prevent those diseases. Metabolic flexibility, right? We're able to shift back and forth. The brain, it has what it needs, and the body has practiced doing it, and that switching is really important. Inflammation, we have an improved insulin sensitivity because we have a reduced inflammation, and an inflammatory response that is controlled. It's utilized in the correct way. And what we see now is neuronal protection. This is actually protective. So we see an increased integrity in the neurons and a resilience against stress. And this actually includes BDNF. So many of you who listen to my mental health talks will know that brain-derived neurotrophic factor, BDNF, is linked to many different mood disorders and mental health generally. We can promote it with physical activity, we can promote it with a healthy diet, and we can promote it with time-restricted eating. So this is another mechanism by which you can understand how fasting can promote enhancing brain health and resilience. Generally, what are the pros and cons, right? We're talking about reducing inflammation, metabolic fastability, autophagy. Here it is, may prevent neurodegenerative disorders. Big old may on that one, right? That this is early research, but it's one of those things that there's pretty low risk, so it may be worth trying. We know about the insulin sensitivity, which, by the way, may be one of the mechanisms that's helping support this neurodegenerative diseases. So there's certainly something here to investigate. The cons, I already said it's not for everybody. There's the potential for nutrient deficiencies, particularly in older individuals where, for instance, protein intake needs to be higher and is often insufficient. So now if you are having a more limited window to take all that protein in, it may be difficult to get all those nutrients in. But perhaps having a less restrictive window could overcome that. There is the risk of overeating post-fat. Some people have kind of a rebound eating after the fast, so that's something to be aware of individually, but also as a clinician. Possible initial discomfort. That's definitely true, but I will say there's also often some long-term benefits, and it does require strict adherence. Okay, so here what are your practical takeaways? So the big principles from this is that you want sun-aligned eating, right? The sun's out, it's good, you're good to eat. A consistent eating schedule, that regularness of eating allows your body to be on a consistent schedule and a rhythm that it will be very happy with. Early caloric intake, I didn't even need to make this point because Dr. Peterson made it for me, which is fantastic. Avoiding late night meals, it's kind of the flip side of that, right? You don't want to be eating right before bed because that's when you're supposed to be resting and repairing, not digesting. And then having a good overnight fast, right? You're mostly going to be sleeping during this anyway if you're getting your 7 to 9 hours. And so hopefully this is just something that you can kind of do for an extra hour or two than you are already doing. In conclusion, fasting works not just because of caloric restriction. It's because it synchronizes our biological rhythms. And it's really important that we eat in an aligned way for many different things, including our metabolic and immune health. And aligning our feeding windows with that natural circadian rhythm appears to be a powerful tool for health and longevity. So there is much more research needed, though. There's some novel approaches here I suggest that I'm not going to get into because I'm short on time. But I do want to just, we aren't there yet. We don't have all the answers, but I just wanted you to see some of these mechanisms so you can understand maybe why TRE could be a good tool. And of course, I have to put a plug in for the Integrative Medicine podcast, which those of you who don't know about it, is freely available anywhere you listen. And with that,